So today we are going to discuss about disposition of xenobiotics. That is, once you are exposed to xenobiotic, what course it takes inside our body. And it is a kind of study of toxicokinetics. What happens to toxic substances when you are exposed to it. So what happens to it? So in this there are four phases which toxic substance undergoes. One is absorption. Second one is distribution, biotransformation, elimination or excretion of toxic substance. So we will take up absorption first. It is a transfer of xenobiotic from the site of exposure into the systemic circulation. During this process, xenobiotic cross body plasma membrane and enter the bloodstream. So once the xenobiotic is in the blood, then we call it as the xenobiotic is being. So to get inside the blood, it has to pass through the plasma membrane. And as we all know, the structure of plasma membrane is consisting of the polar head having phosphatidylcholine, serine, ethanolamine, and inositol. And it has a glycerol backbone which is attached to a non-polar fatty acid. These are the two tails which are being present. And the lipid bilayer looks like this, where two layers are stacked over one another with a hydrophilic surface on the outer side and hydrophobic tails on the inner side. And only the hydrophobic substances can diffuse across it because it has a hydrophobic tails present in it, and hydrophilic substances will not be able to diffuse across the plasma membrane. So this is one of the limiting factor of absorption of hydrophilic substances. So let us see from where the xenobiotic has to, what all distance it has to travel. It has to move through absorption from the external membrane barriers, which include skin, GIT, lungs, and then it has to go into the bloodstream, and from the bloodstream it has to be distributed, and during its distribution, whichever tissue it has affinity for, it gets deposited. They forms the storage depot or sinks, and from the blood it goes into various cells, especially in the liver and kidney, where it is metabolized. There are two phases of metabolism, phase 1 and phase 2, and there are many more reactions other than phase 1: oxidation, reduction, hydrolysis. In conjugation you have glucuronidation, glutathione conjugation, etc. And then it is excreted by the kidney and liver, and even also through lung, saliva, sweat, and so the various cell membrane, as we discussed, it has to pass through is from the environment. If it is your skin, applied onto a skin, or it is through any of the mucosa, gastric mucosa, or it could be lung mucosa. It could be any of the site of exposure, lung mucosa or your dermal skin or whatever. It passes through the lipid bilayer into the interstitial fluid. From there, it passes through the lipid bilayer of the capillary, gets inside the blood vessel. Once it is inside the blood, it binds to the plasma proteins. Then it leaves the plasma protein and then goes inside into interstitial fluid by another plasma membrane. And from there, it enters inside the target cell, where again it passes through the lipid bilayer, and there it gets metabolized and all. And if it is metabolized, some part is metabolized, some part is not metabolized, it is excreted out in the intraorganelle fluid, where it has its target site, some of the enzyme, or it is eliminated by your bile or by kidney, etc. So the physical barrier of absorption, basically it includes the plasma, the interstitium, and the intracellular fluid. So from the blood via capillary, it enters inside the interstitial fluid, and from the interstitial fluid, it goes into the intracellular fluid, where it has its target site. So interstitial fluid and the plasma forms the extracellular fluid, and within the cell, the cytoplasm that we call it is called as the intracellular. So there are various factors which affect the absorption. So there are factors which are related with the chemical, and there are factors which are related with the site of exposure. Factors related with chemical are the physiochemical property of the xenobiotic or a toxic substance, concentration at the absorption site of the toxic substance, and the physical form of the toxic substance. The factors related with site of exposure, that means which is attributed by the body and the tissue or the organ. It is the blood flow, surface area, and the permeability. Let us first take up the factors related with the chemicals. So the first one is a physiochemical property, and in this there are three main properties that play important role. One is the molecular weight. If it is small in molecule in size, it is readily absorbed. Somewhere around 600 Dalton molecule, even if it is hydrophilic, it will be absorbed. Hydrophobic to definitely would be absorbed because hydrophobic substances are readily absorbable. Lipid solubility plays very important role. More the lipid solubility increases the absorption. It is determined by the lipid water partition coefficient or log P. And if a compound has a positive log P, it is highly lipid soluble, it will be readily absorbed. And it has a negative log P value, it is water soluble, it will not be absorbed very. Then another important feature is that of the ionized form. If at all a molecule is weakly acidic or weakly basic, that is, it can exist in the ionic and the non-ionic form, then the ionic form usually has low lipid solubility, thus does not cross readily the lipid domain of the membrane. The degree of ionization depends upon the ionization constant, which is pKa for organic acid and pKb for organic base, and on the pH of the solution. So the pKa and pKb is being determined. It is nothing but a pH at which a weak organic acid or base is weakly ionized, is 50% ionized. So this is called as pKb. Third one is the concentration at the exposure sites. Generally, if there is a high concentration, more absorption would take place. And physical form, solid substances are absorbed slowly as compared to because they have to be dissoluted first. So if at all you give a drug which is already in dispersed form, then it would be absorbed. The factors related with the site of exposure are blood flow rate at the site of exposure. Higher perfusion results in higher rate of absorption. More is the blood flow, more is the rate of blood flow, more the absorption would be there. And on surface area and permeability, there is a linear relationship. More is the surface area, more absorption. Now let us take up the absorption across different sites. So let us first take up the absorption of toxicant. So this is the main route of exposure, oral route, and it is the most important absorption site and important for food and water, contaminants and drugs. Absorption of xenobiotic can take place along the entire GIT. To enter the body, the GIT chemical must pass through GIT lining and capillary membranes before entering the blood. So these are the various sites. Mouth and esophagus, very little absorption takes place. pH of the stomach is 1 to 3, so weak organic acid are likely to be absorbed over here. Small intestine where the pH is 7 to 8, greatest absorption takes place, surface area is 200 meters square, and the rate of absorption increases with the residency time. The residency time of a xenobiotic in here depends upon the intestinal motility. If intestinal motility is less, the residency time would be more and more of the xenobiotic would be absorbed. Rectum and colon, again the absorption. Now let us take up what are the factors. So this was the physiology of the various parts of the GIT tract. So first is organic acid and base xenobiotic. If at all the xenobiotic or toxic substances is organic, acidic or basic in nature. So the toxicant is organic acid or base, it tends to be absorbed by simple diffusion in the part of GIT where it exists in its more lipid soluble form or in the non-ionized form. So basically the mechanism it uses is called as a passive diffusion, that is movement of xenobiotic from its higher concentration to its lower concentration. So wherever it would be present in the non-ionic form, at whatever pH, it is more likely to be absorbed from there only in a passive manner. So a weak organic acid is present mainly in the non-ionic, which is lipid soluble form in the stomach, and predominantly ionized form in the intestine. So weak organic acid are absorbed more readily from the stomach than from the intestine. So this is the Henderson-Hasselbalch equation, which decides about which concentration, at what pH the concentration of the xenobiotic will be more in the non-ionic form as compared to the ionic form. So it is the pH of the medium which affects the ionization of the xenobiotic. So weak acids, they are best absorbed in the stomach because they will be present more in the anionic form. Weak bases, they will be best absorbed in the intestine because they will be present more in the anionic form in the intestine. The second factor is interaction of xenobiotic with endogenous transporters. So there are a lot of transporters which are present, and some of the xenobiotic, they compete with that of the endogenous molecules to pass through using the same transport system to go inside the cell. So one of the example is 5-fluorouracil, which is a toxic substance and also a drug. It is absorbed by pyrimidine transport system. Thallium, it is absorbed by the iron transport system. Lead, which is again a toxic substance, is absorbed by the calcium transport system. Cobalt and manganese, they compete for the iron transport system. Then third factor which influence the absorption across the GIT is the role of transporters, which are present, which have role both in uptake as well as in the excretion. So there are large number of transporters which are expressed in the GIT tract, and they significantly influence the absorption as well as excretion of the xenobiotic. Most of them belong to the SLC family, and they are specialized, they are present primarily in the intestine, in the apical brush border, and they are responsible for taking up the xenobiotic from into the enterocyte from the lumen. Enterocytes are nothing but the intestinal cells. And in humans, there are organic anion transporters present, as well as the organic cation transporters present, which are called as OAT and OCT. There are certain efflux pumps also present, like P-glycoprotein and MRP2 and VCRP. They are responsible for excreting the substrate back into the lumen. And there are some other, there are other like P-glycoprotein present in the intestine, increases from the duodenum to colon. This is their number that is being present. MRP2 expression that is present in the duodenum decreases to undetectable level in the ileum and the colon. And VCRP is found throughout the intestine and the colon. So this is the layout scheme as to how the organic anion and cation transporters present. So this is the lumen, that is where your food comes in. So your xenobiotics also comes in. This is the enterocyte, and these are the transporters which are present on the enterocyte. So they are showing over here, but it is present on the enterocyte. So organic anion transporter, organic cation transporter absorbs the weak organic acid and organic bases, provided they are present in the unionic form. And whatever that is being absorbed, then it can be absorbed into the blood by means of MRP3, another transporter. And there is high chance that the drug or the toxic substances which is absorbed through this transporter can go back into the lumen or excreted back into the lumen by MRP2, PCRP and P-glycoprotein. And there are some transporters which are responsible for transferring the xenobiotic from the enterocyte into the blood as well. Okay, so the journey of xenobiotic, if we say absorption across the intestine, involves movement from the lumen into the blood. So this transporters play very. Then the second factor is interaction of xenobiotic with endogenous transporters. So there are a lot of transporters which are present, and some of the xenobiotic. So here, if at all we take the drug or any xenobiotic through our oral route, and it passes from here into the stomach, and from the intestine, from the stomach it goes into the intestine. From the intestinal lumen, there is a capillary network which is then merging into the hepatic portal vein. So these xenobiotics, which are shown in the form of red dots, they are taken up and reaches the liver, where the liver metabolizes it, and after metabolism, out of many of them which are being absorbed, only few of them in the parent form is going into the bloodstream, so which is a general systemic circulation, and then it goes into the various body organs. As a result, whatever drug that we have ingested, only little bit of it goes into the systemic blood, because much of it is getting metabolized in the liver itself. So this is the first pass. So to get inside the blood, it has to pass through the plasma membrane. And as we all know, the structure of plasma membrane is consisting of the polar head having phosphatidyl, choline, serine, ethanolamine and inositol. And it has a glycerol backbone which is attached to a non-polar fatty acid. These are the two tails which are being present. And the lipid bilayer looks like this, where two layers are stacked over one another with a hydrophilic surface on the outer side and hydrophobic tails on the inner side. And only the hydrophobic substances can diffuse across it because it has a hydrophobic tails present in it, and hydrophilic substances will not be able to diffuse across the plasma membrane. So this is one of the limiting factor of absorption of hydrophilic. Then this first pass elimination plays a very much important role as to how the drug has to be given clinically. So those drugs which have low first pass elimination, that is, they are not metabolized greatly by the liver, like phenobarbital, tolbutamide, pindolol, they can be given orally very comfortably. Those with an intermediate first pass metabolism can be given orally, like aspirin, etc. Slightly more dose can be given. But those drugs which are having undergoing extensive first pass metabolism, then some of them should not be given orally, like lidocaine, hydrocorticosterone, or testosterone. And those, if at all need to be given orally, has to be given in a very high concentration because most of them undergoes first pass elimination, like morphine, pethidine, salbutamol, etc. So the various cell membrane, as we discussed, it has to pass through is from the environment. If it is your skin, applied onto a skin, or it is through any of the mucosa, gastric mucosa, or it could be lung mucosa. It could be any of the site of exposure, lung mucosa or your dermal skin or whatever. It passes through the lipid bilayer into the interstitial fluid. From there, it passes through the lipid bilayer of the capillary, gets inside the blood vessel. Once it is inside the blood, it binds to the plasma proteins. Then it leaves the plasma protein and then goes inside into interstitial fluid by another plasma membrane. And from there, it enters inside the target cell, where again absorption. This is another example of showing enterohepatic circuit. Seventh factor is interaction of two xenobiotics. Two metals can interact with one another. Like cadmium decreases the absorption of zinc and copper. Calcium decreases the absorption of cadmium. Magnesium decreases the absorption of xenobiotics across. Then this first pass elimination plays a very much important role as to how the drug has to be given clinically. So those drugs which have low first pass elimination, that is, they are not metabolized greatly by the liver, like phenobarbital, tolbutamide, pindolol, they can be given orally very comfortably. Those with an intermediate first pass metabolism can be given orally, like aspirin, etc. Slightly more dose can be given. But those drugs which are having undergoing extensive first pass metabolism, then some of them should not be given orally, like lidocaine, hydrocorticosterone, or testosterone. And those, if at all need to be given orally, has to be given in a very high concentration because most of them undergoes first pass elimination, like morphine, pethidine, salbutamol, etc. So there is something like absolute bioavailability. That is when the systemic availability of the orally administered drug is calculated in comparison to the intravenous administration. It is called as the absolute bioavailability. That is, you have given an intravenous dose and you have given an oral dose and then see what is the amount that is reaching the systemic. Absolute bioavailability can be calculated by total amount of intact drug that reaches the systemic circulation following non-intravenous administration, like oral rectal, with the total amount that reaches the systemic circulation after the venous injection. Extravascular administration of drug includes oral, rectal, nasal, subcutaneous. Intravenous includes standard or reference doses to compare the systemic bioavailability. When drug is administered intravenously, the administered dose will directly enter the systemic blood, will not face any barrier, so its bioavailability is considered as 100% and absolute bioavailability expressed as percentage. So with respect to whatever is reaching through oral route, we calculate what is the absolute bioavailability of that drug through that particular. Relative bioavailability, when the systemic bioavailability of drug after administration is compared with the standard of the same drug, is known as relative bioavailability. Relative bioavailability can be calculated by comparing the plasma drug concentration time curves and area under the curve after the administration of two different formulations of the same compound, like capsule versus tablet. So you have a discipline capsule and you have a discipline tablet, and you give it through the desired route of administration and see what is the systemic bioavailability by plotting concentration time curve. Relative bioavailability is the systemic availability of the drug from a dosage form as compared to the reference standard given by the same route system. Then third factor which influence the absorption across the GIT is the role of transporters, which have role both in uptake as well as in the excretion. So there are large number of transporters which are expressed in the GIT tract, and they significantly influence the absorption as well as excretion of the xenobiotic. Most of them belong to the SLC family, and they are specialized, they are present primarily in the intestine, in the apical brush border, and they are responsible for taking up orally. Then bioavailability is high for some of the drugs like acetaminophen, amoxicillin, diazepam, because it reaches unchanged form in the systemic circulation. Whereas bioavailability of some of the drugs is said to be low because they undergo first pass metabolism like morphine, lidocaine, propranolol, etc. So bioavailability refers to the fraction of administered dose of the unchanged drug that reaches the systemic circulation. Features of chemical compound that makes it likely orally active drug in the humans is dependent on the Lipinski rule of 5. So a drug is said to be orally active. That means sufficient amount would reach the systemic blood would be based on this Lipinski rules of 5, which is it should not have more than 5 hydrogen bond donors, not more than 10 hydrogen bond acceptor. Molecular weight should be less than 500 daltons. Octanol water partition coefficient should not be greater than. So, uh,